Thanks very much, Sipo, and welcome to everybody. There are only two things that fill a, um, a seminar room or a lecture theatre. One is if you're under threat or if you're under financial threat. So the reason that there's so many people here probably is because, as Tari asked me, are they here for you or me? I said, they're here for neither of you. They're here, neither of us. They're here for Hantavirus because there's a perception of threat. Okay, so that ship has sailed, and that title, that is a saying, obviously, the ship has sailed, it's left, it's gone, it's too late. But there's an interesting question about this, I guess, because part of this whole outbreak relates not only to the initial transmission event, but how the case that died on the boat was handled, the index case. And I think we can come back to that. It's quite an interesting question of what you do when that happens, and I'll explain why that's important. Okay, so I'm just going to take, before going into the actual outbreak, I'm going to talk a little bit about the virus itself and a little bit about the clinical disease, transmission, and other things. So, and apologies to the virologists in the room and online in advance if I miss anything out. But hantaviruses are part of this family, the bunyaviruses. This is just the bunyaviruses of medical importance. There are many, many more bunyaviruses than this in the classification. And the hantaviruses are also hantaviruses. They are predominantly transmitted by, as we'll talk about, from rodent reservoirs to human. But as we will discuss, the Andes virus, which is causing this current problem, is the only one known at present to transmit person to person. A number of these Bunyaviridae in different groups will be well known to us, like Crimean hemorrhagic fever, for example, or Rift Valley fever. We're going to be concentrating on hantaviruses. And really, there's sort of two major geographic distributions of the hantaviruses that cause two very different diseases or infections. There's the Old World, which is Europe and Asia, that really you see hemorrhagic fever, renal syndrome, and the New World, America, particularly South America, and so some North American viruses that you see hantavirus pulmonary or cardiopulmonary syndrome. So the distribution is pretty stark, and it really does relate to the vectors, the primary hosts. So humans are, of course, incidental hosts. So the primary hosts are rodents, virology, but hantaviruses have also been found in some bats as well. So the first sort of person-to-person transmission cluster and outbreak was seen in the Bolson region in Argentina. Until then, there hadn't been any evidence of person-to-person spread of hantavirus in the New World. And this was in 1995, and the first sort of 15 or so, 12, 15 cases are cases from this outbreak. And what's clear is that there was a lot of contact between different people, and the epidemiology suggested that it was really close contact. So it was especially people who were living together, marital status, boyfriends, girlfriends, etc., or people who shared close contact through travel. And the comparative sequence analysis of the 16 cases sequenced this hantavirus, Handivirus, EpiLink 96, which is the same, as far as we know, the same as the current outbreak. This was the first transmission. The largest Andes person-to-person event was in this area, Epoine, where there were over 30 epidemiological linked cases, and with a case fatality rate of just around a third. And the sequencing and analysis done shows very tight clustering of these patients within this cluster analysis. And again, there's the epidemiological links. And it linked to what was felt to be three sort of super spreader events. These slides are actually this, it's not all it is actually from a WHO emergency consultation that's actually going on as we speak. And Gustavo Valencios from Argentina was giving this talk and showing this clustering around three particularly so super spreader events in the social networks. So person to person was known, and the question then comes as how was it transmitted, and particularly when was it transmitted? And so these clusters, there were actually four clusters, but I just showed one and two. Again, show very much how this was person to person. In cluster one, there was a guy who was a vet. He worked away from home, came back on the weekend, and his youngest son slept in the bed with him while he had just started symptoms, so in the prodromal phase. And unfortunately, this child was infected, and the last contact with his father was 24 days earlier. And so this does suggest prodromal transmission. And in the fourth cluster, actually, without going through this, there was three cases, and again, clustering and close contact. So a guy fell ill after being in a meeting, was went back on the flight, and then shared a taxi with the guy who was sitting next to him, who had helped him off the plane when he became unwell on the plane. So he started having prodromal symptoms just the day before. And then another contact of that second person was involved as well as one of the cases of the genotyping in both these clusters, as shown in homology. So again, this suggests that the prodromal phase was where this transmission event started. And without going into too much detail, what these sort of studies in mouse studies of both infection with Hantavirus virus in rodents and in mice, in rodents and in some of the human cases after post-mortem, show basically that there's an enormous amount of the antivirus in the salivary glands and in the salivary ducts, leading out to sort of egress. And so this is particularly in the prodromal phase, and the thought is then that the infection in the prodromal phase in particular is relates to sort of large droplets from salivary, because the patients weren't, and the mice at that stage weren't coughing. Incubation period was again pretty well defined. We hear up to 42 days in these studies. Not sure how we move this. Okay, maybe we do that, correct? We don't do that, maybe we do that. Yeah, so this is plus 20 patients from Chile, and they did the epidemiological work. Basically, it was 7 to 39 days of very well-defined Chilean Andes virus outbreak. So just a little bit now about the two infections, the diseases that they cause. So the hemorrhagic fever with renal syndrome, so is mainly seen in Europe and in Asia. If patients survive and go through all the stages, because they can die early on, particularly in the hypotensive phase, from shock. The initial toxic phase after the incubation period is relatively short, but they present with fairly non-specific symptoms initially, just as with so many infectious diseases, and sort of fairly undifferentiated fever, but go on to experience rash, experience conjunctival, injection or suffusion, and their laboratory findings show a progressive thrombocytopenia, an interesting leukocytosis with left shift, but without toxic phenomena and atypical lymphocytes. Then there's the shock phase, and if they are resuscitated through that, they go into shock, they go into then a series of events in the uric phase, where they enter hemorrhage as well. There's a pneumonitis complicated by pulmonary edema, and then a variable duration of polyuria for the uric phase, and the recovery can be very prolonged, really take weeks and months, and they're very difficult for these guys seemingly to put back weight, particularly in terms of asthenia. So what we're dealing with is the old world, the new world, which is the Hantavirus pulmonary syndrome, or Hantavirus cardiopulmonary syndrome, sometimes said. Again, after the incubation period, there's a slightly more prolonged prodrome, where fever may be acute in its onset. Myalgia, again, as in with the HFRS, is also an early symptom, and so too is abdominal pain and GI disturbances. Then the cardiopulmonary phase, which can be very rapid and progressive. So it really starts with cough and dyspnea. They have very markedly increased leaky capillaries. I'll show you the endothelial system being targeted. And because they're losing a lot of fluid, they go through typical hemoconcentration, they get an increased hematocrit with low platelet count again, and the respiratory compromise continues and progresses with shock due to vascular permeability. If they survive, they go through a diuretic phase and then into convalescence. So the pathogenesis is largely relates around this endothelial cell damage and the junctions of the endothelial cells, and a lot of different parts of the immune system impact on this. Dendritic cells that are there to sample the environment, take in pathogens, present them to the adaptive immune system, obviously allows the virus to migrate around the body, so a lot of different systems can be involved. Neutrophils that are stimulated, produce IL-8, and IL-8 is also then recruits further neutrophils. You get this sort of meshing together of the neutrophils called netosis and pro-inflammation. And also there's the elevation of this HLA-E expression, which activates the NK cells. So from the innate immune side, particularly, you get this special neutrophil response and a lot of increase in endothelial cell damage and capillary permeability. And then on the humoral side, there's this pentraxin related protein 3, which is a humoral PRR pathogen recognition receptor activating complement cascade, which is also stimulated through complement, again driving this endothelial permeability, and that's why explains a lot of the presentation of this syndrome. I'm not going to spend long on diagnostics. It's just relates obviously to the infection kinetics. This is largely for the this figure relates to the hemorrhagic fever and renal syndrome, but it's the timing of the viral load and the IgM, IgG is similar. Virus can be detected by by PCR, in the very early phase, but as with so many of these infections, because they don't really present until later, that's missed. But then IgM and IgG, the platelet counts, are pathognomonic, nor is the creatinine. So really it's an issue of PCR and serology. One of the clinical sort of bedside manifestations is this has been looked at as a sort of marker of severe early hantavirus infection when they become symptomatic, which is this issue of hemoconcentration from the cytopenia, the left shift, and also the lymphocytes increase in the early immunoblasts. And the sort of sweet spot on the ROC curve comes about 4 out of 5. So this has been used as a screening test for patients being admitted with potential hantavirus pulmonary syndrome, and has been used in community settings and in hospitals to very good effect. So what about treatment for hantaviruses? Well, we're not anything new. The ivermectin acolytes and the apostles are back, and even in South Africa there's a question again about ivermectin. Ivermectin plays no role. What do we have? There is some evidence in animal studies, in lethal challenges, for ribavirin protection in, I think it's a Syrian hamster model. And that shows that 200 higher doses seem to be fatal. And again, the sweet spot is probably around 50 milligrams per kilogram, which does give some protection if you start immediately, but even started later in the course, so day 6 to 14. There has been a systematic meta-analysis of the effectiveness of ribavirin, both in humans and in animals, in the 2 different types of presentation of hemorrhagic fever and renal syndrome and the HPS. But you will appreciate that the numbers in the studies of humans are extremely small, and the main finding that there may be benefit of ribavirin in Asian old world antiviruses came from a single study, which was from China and hasn't been reproduced. It was back in 1991. So ribavirin still looks as if it's probably the best candidate we've got at the moment. However, there are a number of different options looking at people are looking at in terms of therapeutics. There has been some in vitro work around ribavirin and favipiravir that isn't being looked at. But a number of these other areas are very much, most of these areas are very much within research rather than development phase. And so there aren't particularly at this current stage, there aren't therapeutics that are really going to be good against hantaviruses. Vaccines, obviously, are the other part that's being looked at now, again, very rapidly. This is a menu of different types of vaccines, and I'm not going to go through them, but probably the most interesting vaccines at the moment. I mean, there is the RNA vaccines. There's funding issues with that and questions whether we would be able to step up RNA vaccine production, but the DNA vaccines seem to be the potential focus. And one of the problems was that there wasn't a good animal model, because most animals are, with the Andes virus in particular, are infected asymptomatically. But in this study with the Syrian hamsters, if you immunosuppress them, then they present almost identically to what we'd expect in humans. So they've now worked out the animal model, and the safety and immunogenicity of a DNA vaccine has undergone phase 1 study, which was successful. And so I take it now, because of the prevailing outbreak, there's going to be an increase in interest in this, and hopefully we'll be generating antiviral vaccines, not just for Andes, which can be human to human, but there's a major burden of disease, particularly in Chile and Argentina, which needs to be addressed. So in terms of outbreaks, the first time I became aware of hantaviruses was in 1993. So this outbreak of what became Sin virus happened in a place that was very close to my interest and heart, because this is in the Four Corners, which are incredibly beautiful states, 4 states in America, New Mexico, Colorado, Utah, and Arizona. And I spent many years in holidays and things here because my father lived in New Mexico. And he was, apart from being a poet, he was also an anthropologist who worked a lot with the Navajo tribe and the Hopi tribe, particularly the Navajo around New Mexico. And this outbreak started in Navajo Reservation. The index case had it actually been an index case, the first that was identified, was this young guy who was 19, who was a competitive marathon runner, completely well, who, on the way to his fiance's funeral, who was 21 and also a marathon runner, who had died 2 days previously, he collapsed and was taken to the Gallup Indian Reservation Medical Center. And there was this big question mark about why these 2 people who were fit and well, competitive marathon runners, should die of really extremely aggressive and progressive cardiac pulmonary failure. And that led to an investigation. CDC became involved as well. And they, in the end, had 33 cases, all within the Navajo villages and reservations. Interestingly, and as history repeats itself so often, the initial attempt to name it after the place that it was first sort of seen in Muerto Canyon obviously did not go down well with the local tribe, Navajo, and that was therefore changed, and Sin Nombre means no name. So Sin Nombre was the name. For those of you who are film buffs and have seen Vacation, this is from Vacation, because every night couples think it's interesting to go and do strange things on the Four Corners, so you can be in Four Corners states at one time. They tends to get the different states more enforcement. See the movie, it's much better. Okay, so that was so from 1993, and then so when I was at Cambridge from 96, when I went on to the wards to 2000, I really cheesed everybody off by trying to find hantavirus in Cambridge, because there was a lot of people presenting with fever and jaundice and renal failure, which were of course leptospirosis, and I was convinced that hantavirus. I never found one, and I hope that Tari finds a Bruce race, because we just get silly. Okay, so this MV Hondius outbreak, let's get to that. So, as you know, the first case was a Dutch man who died on board. He had been with his wife on a prolonged trip around Argentina and Chile. They'd been bird watching. A few days before, they'd been to a sort of dump site where there were a lot of birds. And there were 147 passengers on board from a number of different countries. So the first case became symptomatic on the 6th of April and then died on the 11th of April. So that's case 1. The boat then docked in Tristan and Cunha, and they organised tours, including tours for the passengers to schools. Now, the captain and the crew had not realised that the death on board was anything unusual, because the patient, although not terribly old, had died of a respiratory illness. And nothing twigged. So, of course, there was no quarantine or isolation at that stage. So they went onto the island, and they went to the schools and went to various other tours of the museums and things on the island. And then got back on the boat from the 15th it sailed, and then on the 24th it docked in another island, St Helena, where they removed the gentleman and his wife, also disembarked with 29 other people, and then she flew to Johannesburg, and on the flight her symptoms began. At the same time, another case, case 3, became symptomatic on the ship on the same day. And the next day, the wife boarded the plane from Joburg to Amsterdam and was taken off because she was progressing and basically collapsed. And so she was on board that flight for a number, I think I can't remember how many minutes exactly, but she was on for, she wasn't whipped off. And she died the same day, unfortunately, in hospital. At that stage, 2 days later, case 3 is evacuated to Joburg from Ascension Island, and then case 4 develops symptoms on the ship and dies about 4 or 5 days later. And at that stage, the ship docks at Papavarnavas, and really 3 days later, first thing that happens is that in Zurich, a man who was had disembarked in St. Helena, self-reported as a passenger because he became unwell, and his virus was confirmed, and it was the first to be sequenced. Then 3 more cases were flown to the Netherlands. One of them was the ship's doctor, and there was some epidemiological information coming at that stage. Remember, this is only a week ago. Okay, so a week ago, then there were 2 flight attendants on the Amsterdam flight who were both tested negative, and 2 Singaporeans who tested negative. And basically, case 9, the Hondius went to Tenerife, and on the 10th, I don't know if you saw the film a bit, but British paratroopers parachuters onto Tristan da Cunha. Now, Tristan da Cunha is in the middle of nowhere, so there's no way they would have been quick enough, because a British person who lives on Tristan, who had been on a passenger and got off at Tristan, had developed symptoms, and they were running out of oxygen. So I think 8 paratroopers parachuted onto this island, and it's incredibly difficult to do. And 2 of them were carrying extra people, so they were buddying. So it was really quite something that they did that, and they got there, and he's now on oxygen. And then there was a case 9, 10, and 11. The American case test is inconclusive. It's the only one to be inconclusive. And then there's also a Spanish passenger who was found to be positive in Nijmegen, and 12 of the Nijmegen hospital care workers are now isolated because they didn't comply protocol. So currently, as we speak, in fact, there's one more now. There are 12 countries, and I believe you have general knowledge to tell you which ones they are, who are isolating contacts. And just this morning, from 10 o'clock this morning, some news came through that there was also a New Zealander who is now quarantined in Taiwan. Information. So, in terms of chains of transmission, this is from the ship. This is again from today's briefing or meeting. So this is early data, but it's generated today, and you can see that there have been chains of transmission within the ship. There has been an early release of a modelling paper that has put the current outbreaks replication number R0 at 2.7. Very good confidence intervals. Is pretty much the same, very similar to other Hantavirus outbreaks. But obviously that's over 1, which means that onward transmission is very likely to occur. And the modelling also shows in blue the identified cases, but is showing the difference in the actively exposed cases, and these are the cases that are actually being quarantined and we need to think about. In terms of age and sex, it really, on this outbreak, really shows the issue around the older well, the young elderly, I would say, between 60 and 69. Yeah, so the median age on board. And this is the epidemiological curves. So 11 cases. The case fatality rate at the moment, 3 confirmed deaths. So the case fatality ratio is 27%. And that one in America remains inconclusive. I don't know why it remains inconclusive, but it does. So these were from Gustavo presentation, and just to say that there's, like with COVID, but more, of course, because of COVID in a way, networks of scientists work together. There's already been, there's been a long history of it. Hantavirus research as an international hantavirus society. There's a lot of people, particularly in affected endemic countries, who are working on this. And there's really a multi-network. I'll just highlight that NICD is part of that network, which are doing a lot of sequencing. And they've already sequenced this, done some sequencing, this one from, I think, the Zurich patient, showing that it was a inflammatory Handius virus that's been causing this. Interestingly, Gustavo Palacios was suggesting that technically it's still, as of today, it's still possible that it's not human to human, only because the animal human timeline with the incubation period still could be 42 days. But everything, that's the only sort of questionable evidence. All the other evidence suggests that it is, and we're pretty much at 42 days, and there are human-to-human transmissions. So we think that that's unlikely. But there's a lot of work to have been done in transmission. And that biggest outbreak in Patagonia versus this outbreak, this slide just very prettily shows the difference in the sort of environment that you're dealing with. In Patagonia, you're dealing with arid, rural community, outdoors, a lot of sort of ventilation. Whereas in this ship, obviously, it's very, very different, very humid, very rainy, wet weather, very much confined. Ventilation systems very different. So lastly, I just want to a couple of slides about the challenges the WHO sees for communication. And really, this context is one of those nightmare contexts, because you've got a very complex, very fast-moving situation that always makes communications difficult. As I said, there are 12 countries currently, 13 countries currently isolating people. With air travel, everybody's concerned about potential for spread. And of course, largely because of COVID, there is a huge media interest. But I think the stress point is that we need to remember that these are communities and the importance of the community. To understand what communities' knowledge is of the risks, understand the fear, the stigma. Marty Setron was just at CDC was used to say, after every pandemic that starts with infection, then there's a pandemic of fear. And that is what we're seeing. That breeds disinformation and rumors. So what are people saying? This is an analysis of social media, and it's the people are frightened that we're going back into a pandemic. They're frightened that we're going back into lockdown. There's a lot of misinformation going around. People are concerned about a long incubation period. It's interesting. The reason they think for the long incubation period is that traditionally with hantavirus infections, the viral amount of virus that's transmitted is actually very low. So it takes a longer time for viral replication to get going and to really impact clinically. And also the initial infection of endothelial cells somewhat reduces the ability of the immune system to see the virus early. So that's one of the thoughts about that. And then, of course, the lack of vaccines, the lack of therapeutics to be able to control it. So lastly, one of their immediate priorities, WHO, in terms of communications. People have learned now there's going to be a lot of tracking. There's going to be a lot of information being given out to try and counter people's concerns, which is quite right. And what we learned in COVID was how important it is to learn about people's different countries' experiences and how to deal with things. So that's my side. I'm going to transfer to Tari, who's going to take us through infection control and what to do.
Okay, thanks very much. Thanks for asking me to give this talk. And I just wanted to show this as a start. It reminds us of the days of Ebola, and also for all the people in the room, HIV/AIDS, we have people wear hazmat suits and go and see patients. And I thought this was quite interesting. This is how they transport patients with Hantavirus in this containment unit, and you can see they put their hands through with gloves, and own oxygen goes inside, and it really looks like from a scary movie scene. And interestingly enough, many of these patients are taken in the US to this Nebraska medical quarantine unit. And I thought it was quite interesting because in the US, this quarantine unit is quite advanced. So people that are quarantining, the people that are currently quarantining are able to have their own room, their own Wi-Fi, their own bicycle, etc. But what's more interesting about this is how it actually works. So the air that comes in is clean, fresh air, but the air that goes out also goes through a HEPA filtration. Furthermore, any waste that goes from their water, sewage, whatever is also sanitized. So it's disinfected, in other words. So obviously it's something they don't want we don't want the virus to potentially move out into the rest of the community. And they have 20 of these facilities, but what's interesting is that they also have the ability to ensure that if a patient does develop symptoms, they have ICU facilities over there as well. And Marc has already alluded to this article talking about the outbreak that occurred in Argentina. And what was interesting is the index case was a patient that went to a birthday party, and at that birthday party, a number of patients became symptomatic, and I love the way they said it in this NEJM article, where patient number 2 was a very sociable person and went to other social events while they were incubating the disease, and as you can see, it just spread from there. But from this article, they kind of alluded to that they think that this could be a airborne pathogen such as TB, COVID, etc. Which brings me to my next point about what is the personal protective equipment we should be using. And obviously, it's going to be your gloves, it's your long-sleeved aprons, it's going to be your visor or goggles, and your N95 for your FFP2 filtering. So it's not actually surgical masks because of the risk of it being airborne. And we'll come back to that a little bit later. Now, of note, there are nice guidelines from the NICD, which you can read over here, which were recently released. And it talks about what if you are a contact of a person that has got hantavirus, and they break it up into whether you're a high-risk contact, a moderate risk, or a low-risk contact. And I won't go through everything, but essentially your high risk for us that work in the hospital environment is people that are exposed to respiratory secretions or aerosol generating procedures without wearing appropriate PPE. And we also need to take into account you have your moderate contact, which is indoor face-to-face exposure or shared transport for more than 1 hour in close proximity. So you need to take that into account. A person that, for example, those patients that were traveling on an airplane, you need to think about the passengers that were around them as well. And then low risk is essentially a patient who's got a brief interaction, no direct food exposure, appropriate PPE, casual workplace or social exposure. And as Mark has alluded to, unfortunately, the incubation period is very long. So it's 42 days. And depending on where your risk is, people, where you fit in this high risk, moderate risk, low risk will define where you should be, whether you should be in stuck at home or whether you need to be placed in a medical center such as the Nebraska one that we've already alluded to earlier. Okay, there we go. I also wanted to talk a little bit about the viral structure because from an IPC point of view, it is important to understand whether it's an enveloped or a non-enveloped virus. And luckily for us, it's actually an enveloped virus. And even more lucky for us is that of the enveloped viruses, it's quite susceptible to disinfectants, etc. And just to put into perspective, I like this slide because it describes the microbicidal efficacy. And as you can see, envelope viruses are all the way at the bottom over here compared to your prions, which are right over there. And then you compare it to your non-envelope viruses, it's a lot more susceptible to your microbiocides. And this is a preprint, but I did think it was quite relevant and interesting. So they basically took the Andes virus, and then their control was Hantavirus, specifically the Tula virus, and they saw what would happen to the virus over time and also in the presence of dehydration. And what they showed is that at 37 degrees, your virus actually no longer becomes infectious after, they say, 13 hours. But if you leave it at 21 degrees, you can see it can still remain infectious for quite a number of hours. And even if you go to if you go to 4 degrees, it can remain infectious for quite a prolonged period of time. And then this also brings me on to the next part is what temperatures are able to kill the virus. And one can see from above 50 degrees, and in actual fact, if you hit 60 degrees for more than a minute, you will definitely be able to reduce the infectiousness. And that takes into account what do we do with linen from these patients and their beds and disposable stuff that you actually do need to ensure that you're reaching at least 60 degrees Celsius to try and get rid of this virus. And I'm not going to go through all of these, and I'm going to show it again later, but it's a hell of a susceptible virus to different forms of detergents that you can see over here. And furthermore, I thought this was quite a nice article. I really do like the Journal of Hospital Infection. I think it's a brilliant journal that really does go into quite a lot of detail and has nice articles from an IPC point of view. But this was recently towards the end of last year where they described the stability of Andes viruses with regards to the WHO recommended alcohol-based hand rubs and other surface disinfectants. And one can again see that your alcohol-based hand rubs and your aldehyde-based hand rubs are quite effective at decreasing the infectiousness of your virus. And what I liked about this is the alcohol-based hand rubs that we have in our wards. It depends. Sometimes you get 70%, sometimes you get 80%. But these are the WHO formulations over here. And depending on the amount of concentration of your disinfectant, they show that even at disinfection levels of 30%, you're still able to neutralize the virus. So what am I saying to you? I'm saying to you, if you need to clean the room, I think a good terminal clean with the correct disinfectants, detergents, good alcohol-based hand rub, you should be fine in seeing these patients. And then just for the virologists and the IDE people in the room, I think it's also important to remember that it forms part of the category A infections, which means that when we do need to transport blood samples from these patients, that it needs to go into one of these containers, and what you can see is here you'll put your blood sample inside, And then because it's a blood sample, you actually need to put an absorbent material around it. Then it is sealed, okay? And then you seal it again and over here, and then you put it in this box. And the reason why there's so many different layers to it is that if it falls off the back of the truck, at least it won't be spread into the rest of the environment because you've got your absorbent material, whatever it is, to prevent it from being spread of the disease. And then I went and looked up, there's actually a hell of a lot of organisms that fall into this category A. And even hepatitis B is over there, but this is for cultures only. Then you can see that Hantavirus falls over here as well as your Ebola, etc. And in summary, which is rather, it depends, it can be quite a controversial point over here. I like this article that Marc shared with me earlier today. It was an opinion piece, and the author said WHO should say plainly that Andes virus should be managed as a pathogen with plausible airborne transmission, and that recommendations should start stringent, then ease only if evidence allows. This is a precautionary approach this outbreak demands. And I think, whenever we start off with these new viruses or new infections, not that this is new, but we haven't seen it for so many years and we don't really know about it, is maybe it's not a bad idea to have extra IPC precautions in the beginning. And then once you understand the dynamics of the virus, then you can move across to less stringent precautions. I mean, if we all think about what we did during COVID, we all almost suited up. And then at the end, we were just wearing surgical masks. But maybe COVID N95 were a better point at that time. So, yeah, thank you.
Well, thanks, Tari. I just wanted to clarify one thing at all from what Tari's just mentioned now. So when I was discussing the mode of transmission, the prodromal phase is particularly where you will get salivary transmission, so large droplet. But within the pathogenesis and cycle of hantavirus infection, alveolar macrophages will be infected. And so the issue about whether it's large droplet, small droplet, etc., it's both pretty much. And even during the prodromal phase, I'm sure that you would be able to transmit via a smaller droplet. Whether you can transmit by micronuclei, I don't know. But I think that just reiterate what Tari's saying is, you start off with the worst case scenario, and as we understand more, you go to first like a face mask rather than a ventilator and whatever. And it's always interesting that to see what sort of the level of IPC and containment cases or contacts are being dealt with in all these different countries. Yet there isn't firm instruction on using the same sort of protection as that. And so just to come full circle, the question you first posed, it was around whether, having identified a patient who died on the ship and quarantined and taken interventions, or made interventions at that point, would have changed what we're seeing. And it's very difficult because cruise ships in particular often have an older population. There are often people with comorbidities that are often treasured. It's the patients where it's unexpected, just as it is whenever we see a patient, an unexpected death. That's got to raise questions, and particularly for cruisers that are embarking and disembarking around endemic areas